Well, thank you very much, Brenda. I hope I don't come across too mad as the scientist today. Okay, so I should just give a little background. Although I'm a scientist and I'm a basic scientist, I work at the Francis Crick Institute in London. Actually, interestingly, Mary Shelley was born just a few hundred meters from our new institute. It doesn't mean we picked up all her thoughts. So I'm a basic scientist, but I have been involved quite a lot in discussions at the levels of ethics, regulation, policy, etc., in this topic as well as other quite contentious topics. So I don't have a pointer that's going to really be of any use, but if I need to, I can try and use this. So, of course, humans have been manipulating genes in animals and plants for a long, long time, so at least 12,000 years, with the domestication originally of dogs from wolves, and then and various other animals, and of course many plants. Human genes and genomes have of course also altered over the same time. Due to selection for specific traits, and there's lots of evidence to suggest that. So that can be changing in response to changing climates or changing diet. So lactose tolerance, for example, is a very good example of that. But of course, this has been due to so-called natural, our own changes have been due to natural rather than artificial selection. And of course, all of this went on without any knowledge of genes and of DNA. So what I want to do is just give you a very brief history of our ability to genetically alter animals, and this is really going back to the origins of genetics in science. So of course, Gregor Mendel discovered genetic inheritance 1865 or thereabouts by his initial experiments on pea plants. Nucleic acid was discovered a little bit later, and it was thought maybe it was the basis of inheritance. It could be the basis of inheritance already back then. Other terms got introduced, including chromosomes and mutation, I guess in the late 1800s. And then chromosomes were thought to be the carriers of genetic material around the turn of the early 1900s. Then we have a period where a lot more information was obtained about genes and genomes, and a lot of that came actually from mutation studies. So this is obviously relevant to what we're going to be talking about because we are talking about essentially altering DNA through mutations. So initially that was X-rays and UV radiation, but then of course later chemical mutagenesis was used a lot. The proof that DNA carried genetic information and not proteins associated with the chromosomes came around by classic experiments done by Avery, McLeod, and McCarty around 1943. And then other clear demonstrations a little bit later in 1952 by Hershey Chase, and that was done with phage, which are viruses that attack bacteria. And then, of course, it was the elucidation of the DNA structure by Watson, Crick, with obviously a lot of help from Rosalind Franklin, in 1953, which gave the structure of DNA and provided the first clear mechanism of how inheritance might work. Since then, there were a lot of studies started, I guess, in the early 1970s, which showed that it was possible to manipulate DNA in microorganisms like bacteria. This relied on a couple of particular important enzymes that were discovered around that time. And then there was the ability to actually sequence, so read the code in DNA which was Fred Sanger, Maxim Gilbert, and others towards the end of the 1970s. Now, almost every new technology since about 1973, so manipulating bacteria, for example, has prompted a debate about the possibility of genetically modifying humans. And of course, in 1978, with the birth of Louise Brown with IVF, the first IVF baby, that debate intensified a lot. The first actual manipulation of DNA in a mammal occurred in around 1981, or a little bit before actually, but 1981, which was making so-called transgenic mice, which simply involved the micro-injecting pure DNA into the fertilized egg. A proportion of eggs took up the DNA, integrated at random within the genome. This is very inefficient and haphazard, but it works, and it works well. In fact, we still use it in my own lab because it's a valuable technique to manipulate mice. It's been used for lots of science since it was first developed a long time ago now. Another method was developed shortly after that, and that was to make use of stem cells that come from the very early embryo, a very early mouse embryo, so-called embryonic stem cells. These can be grown in culture, manipulated in the lab, so you can make a desired genetic change in the lab. And then you can reintroduce these cells back into the very early pre-implantation embryo, so-called blastocyst stage, which is this, which is essentially the same stage that the cells would have originally been derived from. And they will integrate into the embryo and give you a so-called chimeric mouse, which is a mixture of the host embryo and the introduced cells. And you can then breed from that and establish a line of mice, and this is showing one that's expressing green fluorescent protein. So that turned out to be a route to altering the genome that allowed not just integrating things at random, but through careful design of DNA, the DNA you're putting into the cells, and a method of selection, you could find cases where you could specifically target a change to a desired sequence, a desired gene. This is so-called homologous recombination, where the parts of the DNA you're introducing are common to the DNA and the gene you want to alter, and so you get a recombination. But this is a very rare event. It's probably much less than 1 in a million cells, so you have to have a method to select out the cell that you want to find. It's also rather complex and rather inefficient. And again, because you have to go through the stage of making chimeras, it's a long, torturous process to get there. It's been fantastically useful, lots of good science from it, but it's never been a technique that you could really think of applying, certainly not to humans. Then, as mentioned, we have Dolly the sheep, was cloned. This, I'm not going into the methods of this, but basically, you take a somatic cell nucleus, a nucleus from, say, a skin cell, transfer it into an enucleated egg, and then kickstart development, and it will develop into an animal in a few cases. So again, it's a very inefficient method of getting rise to an animal from a body cell. But what you can do, of course, is manipulate the genes within that original starting cell, your skin cell, for example, and then go through this route of cloning an animal. And that is still actually a well-used route to obtaining genetically modified farm animals in particular. So it has a number of advantages because you know what you've got before you start. And even with using the genome editing methods, which are much more efficient, that route is still often used. The success of cloning through the somatic cell nuclear transfer route led Shinya Yamanaka in 2006 to develop this method of directly reprogramming adult cells back into embryonic stem cell-like cells. So these cells now they've arisen from almost any adult cell type and have been reprogrammed back to think that they are early embryo cells. So these again form a way of manipulating genes and then getting an animal back. But of course, you have to go through this route of making a chimera if you really want to do it at the moment. Another route to manipulating genes in animals has made use of stem cell population that's found in the testes of male animals. This is spermatogonial stem cells. So these are the cells that they will divide and they will make a copy of themselves, a process called self-renewal. But then they, of course, can also differentiate ultimately to give rise to sperm, which will then fertilize an egg. It's possible to grow these spermatogonial stem cells in culture and to manipulate them in culture, so you can alter the genes in those cells. You can then put them back into a testis of an animal, preferably depleted of its own spermatogonial stem cell population, and then it will enable you to then effectively fertilize eggs using those sperm that are developed in that testis and get your genetically manipulated animal. So this is just showing a picture of some rats that have been genetically modified that way, again to express the green fluorescent protein. It's also been done with a number of other animals, so mice, rats, and even macaques, so non-human primates. And while I'm talking about using manipulating cells that will give rise to germ cells, you could also, in theory, and this has been done in the mouse, you can start with a pluripotent stem cell, whether that's an embryonic stem cell or induced pluripotent stem cell, iPS cell. It's been shown that you can derive either sperm or eggs, perhaps entirely in vitro, but certainly in combination with in vitro, start off in vitro, and then you put things back into either the ovary or the testis. And so this is another route to manipulating genes in the germline. Okay. So what has driven all this technology as applied to mammals? Well, it's mostly, of course, basic science and curiosity about how genes work and what they do. Ways to follow things during embryonic development. A lot of stem cell biology, we use these techniques for trying to understand the stem cells. Say the role of specific genes in embryo development, physiology, the immune system, etc. Also to try and understand aspects of human genetic disease using animal models. Cancer biology, huge amount of work. And then, of course, as I mentioned, manipulating genes of farm animals, it's had a number of practical or proposed practical uses, how to make them more productive, grow faster, have disease resistance, and things like that. So with each new method of manipulating genes in mammals, the same questions have arisen about the possibility of using them to treat or avoid genetic disease, make designer babies, or practice eugenics in a human context. But it's always been possible to say very clearly that these methods are far too inefficient, far too unsafe to be applied to humans. But at least two areas of science have now opened up these debates again, and which is why we're having this meeting today. So one of them is, in fact, our ability to understand human genes, genomes, and genetic variation through ways of just simply finding out what the genome sequence is. So the the cost of genome sequencing has come down dramatically. The speed has gone up. I mean, it's advanced greatly. So we know a lot more now about genes and genome genetic variation. There's still a question whether we know enough, but we can talk about that. So if we have all this knowledge about our genes, why not use this knowledge to solve some of our own genetic problems? Let's say we've had these methods to alter DNA mammals since the early 1980s, but it was always possible to say that they were just too unsafe to even think about trying. So the other thing that's really driven this new debate is the precise and efficient means of altering DNA provided by these genome editing methods. So genome editing methods, I'm going to talk a little bit about this. If you don't want to understand all the details, you don't have to. Just get the message that these methods are very efficient and precise. So it makes use of DNA repair mechanisms that exist in all our cells. Our DNA is constantly getting broken and has to be repaired. Every time cells divide, the DNA gets broken in many places and it has to be repaired. So there are these different ways of repairing the DNA, and I will touch on that in a short while. And genome editing requires 2 or 3 other components. It requires you to introduce so-called, some people refer to them as molecular scissors. I actually don't really like that term, but just for simplicity I will use it. So this is an enzyme that is able to cut DNA. And actually, what's usually used is one that will cut the DNA to make a double-strand cut, so it cuts both strands of the DNA. That triggers the DNA repair mechanisms. But you need some sort of homing device, a way of taking that enzyme to the right place in the genome. And that can be derived from either DNA-binding proteins, which have been designed in the lab to recognize a specific sequence, or it can rely on an RNA molecule which is complementary to the DNA that you want to target. So it's a nucleic acid. And then if you want to do more than make a mutation, then you also need a template, a DNA template of some sort, which is going to have homology, so have the same sequence, at least part of it have the same sequence to the gene that you want to edit. But then the rest of it has the change you want to make. Okay. So there are several methods of genome editing, and I'm going to mention a couple of the old ones as well as the one that most people are excited about. So the first one that was really developed properly is called the zinc finger nucleases. These are, again, this is a protein elements that you can stitch together to make a protein that's going to recognize a specific sequence. And usually these are designed to recognize a sequence of around about 20 base pairs, which is enough in most cases to guarantee that there's only one site in the genome that in this case that this protein will bind to. And then this is linked to an enzyme, and the one that's usually used with zinc fingers is the FOC1 enzyme. You need 2 copies of this because the FOC1 only cuts one strand of DNA. So you have 2 zinc fingers that are going to bind opposite strands, bring 2 FOC1s and make a double strand cut in the DNA. TALENs is a very similar system, but it's actually easier to use in the lab. And it works again on a modular design of protein elements that will recognize specific DNA sequences. These are being used. They're being used in clinical trials, so we mustn't forget them. They are still important. They have a number of advantages, perhaps. The method that people have got a lot more excited about is the so-called CRISPR-Cas9 method. So CRISPR is a terrible term to have to use because it stands for clusterly regulated, regular interspersed short palindromic repeats. this comes from the function of this system that it was adapted from, which is essentially an immune system in bacteria. So when bacteria are infected by these bacteriophage, these viruses, it's possible that the bacteria, they have this system which can, if they survive the attack, they can recognize now a bit of the sequence from the virus, and they use this system to then destroy the virus if they're infected again. And of course, it's inherited in the daughter cells, and so the population of bacteria gets resistance to infection by the virus. So anyway, the CRISPR-Cas9 system, so the CRISPR part is the RNA guide. So it's an RNA which is complementary to the particular DNA sequence you want to target. And then the other part of the RNA includes a portion which will bind to the enzyme, the nuclease Cas9. Cas9 makes a double-stranded cut in DNA, so that's one reason why this method is more efficient, because you immediately bring a double-stranded, an enzyme that's going to cut both strands of the DNA to the target sequence. It's also incredibly easy to make the RNA guides. In fact, you can just order them. It's so easy. So it's very simple, and it's cheap. I would say. It's a very simple way of doing genome editing. And there are lots of advantages of this. So it's simple to make components, relatively inexpensive. It's relatively simple to introduce these into cells, or, as I will tell you, into early embryos. They're highly specific, highly efficient. You can multiplex, meaning you can target more than one gene at the same time, and that's been shown to work very efficiently. They're versatile, so you can make alterations in DNA, pretty much anything you want to do, from a single base pair up to many thousands of base pairs. You can also make use of an inactivated form of the Cas9 enzyme, so-called dead Cas9, which you then link to other enzymes that can modify DNA in different, or modify chromatin or chromosomes in different ways. And I will mention one of those in more detail in a minute. But for example, you can alter the way genes are expressed, not changing the DNA sequence at all, but just alter the way genes are whether they're active or inactive, by bringing certain so-called epigenetic modifiers to a specific region in the genome. So let's go through a little bit more detail. And I'm sorry if there's too much detail, but so this is just, if you want to just simply make a small mutation in a gene or the coding sequence of a gene, then you can just simply have the CRISPR-Cas9 and your guide, so you have your guide RNA and your Cas9, which will hybridize together on the specific sequence you want to target. You have a double-stranded break in the DNA, and then a common DNA repair mechanism that will try and stick that back together again is so-called non-homology end joining, and I'm sorry I didn't choose the term, but NHEJ, which essentially just sticks the DNA, two broken strands of DNA back together again, but it often makes a little mistake. So it often creates a small insertion or deletion, usually of just one or two or three or four base pairs. So if it's you're trying to inactivate a protein coding part of a gene, if you make just a single base chain, one insertion or deletion of one base pair, or two, that will inactivate it. 3 might not, 4, 5 will, etc. So it's a very simple and efficient way of making mutations in genes. Even though it's making mutations, it can actually be a benefit, and it's being proposed to use this for somatic genome editing, which I'll get onto in the second part of the talk, to treat Duchenne muscular dystrophy. Because often Duchenne muscular dystrophy is due to a mutation in the protein coding part of the gene, but that is divided into many exons, so many separate pieces. And so if you can make a mutation that allows you to skip that one exon, you can then make what is essentially a fully functional protein again. So you can use the CRISPR-Cas9 to exchange sequences, and this is so-called homology-directed repair. So in this case, you start off in the same way with your RNA guide, your Cas9 protein, but now you also introduce a DNA template which hybridizes to its cognate sequence within the gene. And so you have two parts which are homologous, and then the change you want to make is put in the middle, and this seems to be missing a bit, maybe not. So it will recombine in, and whatever's in the middle will now get inserted in place of the sequence that was in the genome originally. You can actually use this to make deletions, quite large deletions if you want. You can use it to make large insertions. You can use it to make just a single base pair change, and you can use it to swap in sequences that weren't there at all. So completely novel sequences can be inserted into a gene in this way. So this is homology-directed repair. Now, the third method I want to mention, because this is relatively new, but actually very exciting, is so-called base editing. This makes use of an inactivated form of the Cas9 enzyme. So it doesn't make a double-stranded cut in DNA, but it's hooked up to another enzyme which is called cytosine deaminase. This chemically modifies one of the bases in DNA, one of the letters C, and it goes via a U step, but then that gets turned into a T. So you can change a base from a C to a T. That creates a little mismatch in the DNA. So you now have a T at the top, which of course can't pair with a G. So there's another DNA repair mechanism called DNA mismatch repair, then detects that mismatch and changes the C into an A. So now you've got proper double-stranded paired DNA again, but you've changed what was originally a CG into a TA. Now, why am I talking about this? It's very simple. You're just changing one base pair. Probably half of all genetic diseases in humans that are due to simple mutations in genes, so-called Mendelian inherited genes, are due to a change in a single base pair. So this method could be incredibly important as a way of either studying those mutations in the lab or repairing them in individuals. David Liu, MIT geneticist who has been working on this, developed this particular method. So the one I just described is on the left here. He published a paper towards the end of last year where he had made a new enzyme that allows you to convert things back the other way. So you can now go from an AT to a CG. He's trying to make other enzymes that allow other changes between base pairs. So these methods could be incredibly powerful. So these methods are now sufficiently, perhaps sufficiently precise and efficient that the old arguments about methods altering DNA being too unreliable and unsafe to use with humans may well no longer apply. And these methods are being used, of course, to model and study many human genetic diseases in mice and other animals, including rats, pigs, sheep, and non-human primates like macaques. In attempts to, they're being used in attempts to modify animals, such as farm animals, for a number of properties, including a source of organs for humans. That's a long way off, by the way. To make animals and plants resistant to diseases. I've given examples there: African swine fever, to make chickens resistant to influenza, cows resistant to prion diseases. They've been used to perhaps improve animal welfare, reduce antibiotic use, in attempts to prove animal and plant characteristics as a source of food, clothing, other uses, and in attempts to control invasive animal and plant species, or disease vectors such as mosquitoes and ticks. This brings up another sort of use of the method, which is so-called gene drive, which I'm not going to talk about. If you thought that was complicated, gene drive is really complicated. But it's these methods are being, say, contemplated for use, for example, to eliminate malaria-carrying mosquitoes, but they're still a long way off from being used. Absolutely no one's contemplating using them in humans, I guarantee you. So I'm going to stop there for a few minutes, and just if anyone wants to ask some specific details before I move on.
Thank you, Robin. Yeah, let's give the man a round of applause, shall we, for that, first of all. Thank you very much. We are going to pause literally just for a few moments, and only to take questions for factual information. I'm going to disallow two types of questions. The first one says, can you give me a course in Human Biology 101 from scratch? If this has just been gobbledygook to you, well, hold on until this afternoon. There's nothing we can do about that. No. On the other hand, if you are already an expert in this field and you want to ask something of minute detail, then maybe you can keep that to lunchtime and ask Robin then. So within those wide parameters, any questions for information? There we go. Yeah. Yep. Just here in the middle, please.
In the middle here.
Right?
Yeah. Sorry.
Just if you put your hand up nice and high so we can see. Thank
and
you.
wait for the microphone's coming. Yeah.
Yes. I would like to know where this research takes place. Is it in the Western world, or is Japan or China involved? So what is the spread of this knowledge? Where's the focus?
The methods are, they were, I mean, I guess the originally developed in the US, but closely followed actually by groups in Europe and South Korea. And they are now worldwide. So every basic research lab, institute or university or company pretty much uses these methods. They spread rapidly. So the CRISPR-Cas9 methodology was only sort of invented 6-7 years ago. It's now all over the world. So, and they are used in, say, in basic labs, research labs, clinical research labs. I'll talk in the second half about potential uses in clinical uses, and by companies for all sorts of different types of science. They are also now, and this gets a little worrisome in some respects, used by the so-called garage science community or biohacker communities. So these are people who want to play at being scientists in their kitchens or their garages. And they're starting to use these methods as well. They can buy kits. So you could buy a kit, order a kit online, and do it at home tomorrow.
But please don't.
Well,
so
you might want to.
Not yet, anyway.
Yep.
Yep.
Thank you for this interesting presentation. I have a question concerning the typical characteristic features of CRISPR-Cas: cheap, efficient, precise. As far as me as a layperson heard about the first hype of CRISPR-Cas, that was the general characterization. But in the last month, one could say, I read also that these general characteristics come under pressure, that it is not as precise as announced, not as efficient as announced. Could you put us more into detail?
So several things. So the methods depend, as I say, depend really on the efficiency of the DNA repair process that's within the cells. Non-homology end joining, the simple one of where you stick it back together and make a mutation, is present in many cells, and it's a very efficient process. The homology-directed repair, where you're making more targeted changes in the genome, is generally a slightly less efficient method. And it's often, you will have both non-homology end joining and homology-directed repair mechanisms operating at the same time within a cell. So if you, for example, our experiments on the mouse, that you can, you're trying to get homology-directed repair, and you get it, but it may be that you get it in one copy of the gene, but the other one has now picked up a mutation due to the non-homology end joining. So yes, it's not, the homology-directed repair is not quite efficient enough in most circumstances. But that can be manipulated, and again, I mean, I could have added more details, but you can use cells at the right phase of their cell cycle prefer to use the homology-directed repair. You can use inhibitors of the other DNA repair pathways. So you can push it to get your homology-directed repair. It's also depends on, because you have to have a DNA template introduced at the same time, and that's very critical, that you have enough DNA template in at the right time when your Cas9 enzyme is going to be active, that it leads to the appropriate repair. So you can manipulate things to make that process more efficient. The base editing, which is this newer method, again, people haven't really worked on to make it more efficient, but certainly in the cell systems that have been published, and I'll show you one result with embryos, it's at least 50%, which is pretty good. And so we think that these methods can all be pushed to make them even more efficient. I mean, some of our experiments, just relying on non-homology end joining in mice, we get 100% of our offspring born with a mutation. So they can be very efficient.
Thank you. One more question maximum. Anyone put your hand up nice and high?
We can talk about the accuracy later, shall we? Because that's the other part of your question.
Okay, Robin, if you move on,
yeah,
please,
sure.
to your second talk.